Thanks everyone for attending my talk, despite that I'm not there in person. Indeed, I'm now in the Netherlands, so for this reason, I'm joining you online. And I want to show you some exciting work in the brief period we have together about working with complex systems in science and addressing this complexity. So to start with, I want to sort of show a bit of a personal angle to the research that I do by showing you a picture from my biology high school book. So specifically, this is a sort of scheme of a human cell as it was shown to me in 2010 when I was learning about biology for the very first time, essentially. And you can see some of the components that are inside. There is a vacuole and there's some mitochondria. And at this point, I found out this is quite interesting, and I thought it would be interesting to study how this cell works and also how it misbehaves, so how disease arises. So I decided to do a bachelor's, but the picture of the cell essentially kept expanding. So here you can see the cell as I saw it in 2017, and there were many more compartments in there doing many different more functions. So essentially, the complexity of the system I was trying to understand increased as I studied. So here you can see another picture. So this is what the cell looked like when I was a master's student in 2019. And you can see many more different structures starting to arise. And then finally, this is a picture during my PhD, where instead of showing a human cell, essentially in the compartment it contains, you can see here that we're showing specifically one species under a specific condition, rather than all cells, to make sure that we can show all of the complexity. So complexity was really sort of very much present in the research I was doing in my studies. And I joined Thomas Knowles as a PhD student to address this complexity and to make progress with the applications that we're interested in. So the different compartments you saw in the previous cell, those are organelles of the cell, the same way that our human body has organs, the cell has organelles, and these perform different functions. And so some of them, they have a lipid membrane around them, so that way they're sealed from their environment. And there's also organelles which do not have these lipid membranes, and they're rather formed by phase separation. So the proteins, the RNA, sometimes DNA, will self-assemble into a compartment that then performs a function for the cell. And then here on the left, you can see a phase diagram, which is how we describe such systems. In the middle, you can see these green puncta inside of cells. So those are the compartments. And here on the right, you can see the same, similar system, but then taken outside of the cell. So why are we interested in all of this complexity? Why bother in the first place? Well, it is because it helps us to understand how cells work. So how does life work? It helps us to understand how to treat diseases. Specifically, neurodegenerative diseases are associated with phase separation. But it also helps us a lot with applications, which is what I'm very interested in, specifically biotechnology applications, food science, material science, and also artificial cells, so that is artificial life, creating life without starting with life. So putting molecules together and creating a life form, which of course has not been accomplished yet, but is a dream of many scientists for the future. And phase separation is seen as a critical tool in this. So I want to show you some of the technology that I've created during my PhD together with others to understand complex systems. And one of them you see depicted here, where instead of studying phase separation by looking at one sample, we look at many samples and vary the conditions. So here specifically, you can see that we change pH, acidity, and concentration of a chosen molecule. And in this way, we can create sort of a map in chemical space and see when phase separation does or does not occur. So when we get two phases and when we get one phase. So this kind of technology can really help us to understand these systems. And what we've done is we've combined this kind of measurement with microfluidics, which is an extraordinarily interesting tool whereby you miniaturize your experiment to the size where you usually cannot see it anymore, by the naked eye anyway. So here you can see a few images of what this process looks like, where we create these really small droplets. And these droplets are then actually our samples, and they're smaller than essentially the width of your hair, of a single hair. So it becomes very difficult to see your experiment, but of course, then when you combine it with a microscope, then you can see your microfluidics. And we combine this then lastly with machine learning to look at all of these images, study what's going on, and then turn all of those samples into useful diagrams that we can interpret. So here on the left, you can see this phase diagram, which shows you when you have two phases or one phase and essentially describes the system we're interested in. But you can see here, N is about 38,000. So that means that the amount of data points that we are able to gather for this graph is 38,000, which is quite a bit higher than you'd be able to do with conventional technology. And because we're able to go to so many samples, we're able to understand more interesting systems. We can study it in more dimensions. So it was quite sort of a shock to, during my PhD, be able to go from measuring about 10, or maybe if I worked very hard, 100 data points a day, to essentially have created technology that measures 10,000 data points an hour with a fraction of the material at the same time. And so here you can see some pictures of me and Thomas Knowles, my PhD promoter, in the lab. And it's really been sort of a very interesting thing to go from having a calculator to having a computer, which you could compare this to experimentally. And the last thing I want to share with you is that these kind of insights into molecular systems really help us to also understand larger things. So you go from insights from molecules to structure to function. So I want to show you one of the structures that we found and studied during my PhD. So here you can see a phase-separated system that then also contains compartments inside, and you can see how this develops over time. And so this has really helped us quite a bit to understand more about how the cells create structure and how we may apply it in the future. So to briefly wrap up, phase separation is a very interesting phenomena for understanding life and treating diseases, and also various biotechnology applications and artificial life. But the problem with these sort of systems is that they're very complex. They depend on many variables, very subtly, sometimes non-linearly. And so this kind of limits the amount of applications that we can work on. And for this reason, I've worked on technology to study these systems. Specifically, I've combined microfluidics, microscopy, and machine learning together into platforms. And in this way, we've moved from understanding how molecules assemble into certain architectures or structures, and are now working on function. Nowadays, you can find me in Eindhoven in the Netherlands, where I work together with a really great team of scientists on the big chemistry group. It's a robot lab, is it also what it's called, where we combine robotics and artificial intelligence to create even more of these platforms to perform different functions. So we automatically study many different chemical systems for many applications. So for food science, coatings, paint, yeah, most of the chemical things you might want to do, we're automating and going into higher complexity. So with that, I would like to thank all of my scientific collaborators, and particularly the Society, for inviting me to give this talk and for awarding me this prize. And thank you very much for your attention, and do reach out if you saw anything of interest. Thank you very much.
Thank you very much, Nadia. Are there any questions from the floor? I have a question to begin with, but there'll be a mic coming around when John. But Nadia, looking at your phase diagrams, and in particular that video, I used to be a materials scientist, and we did things like that, spinodal decomposition, spinodal alloys for ultra-high strength alloys. And then the video of the green things clumping together, that's a phenomenon we called Ostwald ripening in iron carbide precipitates in steel, and that's just perfect. But that's bigly. The the question I wanted to ask is, are there any, with such small samples, are there any interface effects on the chemistry? You said there are 50 odd microns or so.
Mhm.
Does the interface between the green bit and the black bit influence the chemistry, do you think?
Absolutely, absolutely. In fact, it's a really great question, actually. This is something that we've looked at quite a bit, because when you see these kind of structures, the main thing you're varying is the interface area. So the first thing you start to think is, do cells use this, or can we use this for something? What we see is that molecules can locate themselves depending on their properties in different parts of this structure. And some molecules, they like to concentrate themselves specifically at the interface. This is called absorption. And we see both that this happens in biology, so in cells, and this is something that we're working on using, actually, yes.
Okay, thank you. A question from the floor here from George.
Yeah, that's a fantastic talk. Thank you very much. How well can you quantify with traditional microscopy, as you show here? Do you need to resort to confocal? Would that give you an advantage?
Yes, this is a really great question. So the video that you saw here on the screen, that was confocal. And in the past, these measurements we've done with a wide field fluorescent microscope. And one of the reasons that I'm now in Eindhoven doing research is because now I've combined this platform with a confocal microscope. So yes.
Any more questions from the floor? No. Okay, Nadia, thank you very much indeed for a very interesting talk. Good luck with the big chemistry. I've not heard of that before. That sounds fascinating stuff. I hope it all works out really well for you in Eindhoven.
Thank you very much, John.